Hey guys, welcome back to Young-Splaining Cellular Respiration and Photosynthesis. This is video 2, where I'm going to go step by step through the processes of glycolysis, and I'm going to go back through the citric acid or Krebs cycle, as well as electron transport. So we're going to look at this first 5 steps of glycolysis using glucose and the last 5 steps using glucose. Remember that the first 5 steps is called the energy investment phase, so it's where I am going to actually use energy. Now, when I go through this, I'm going to tell you some things about these substrates and enzymes, because I think if you know what's going on, it'll help you remember this better. Okay, so first things first, glucose is our initial substrate. And when I say substrate, that's like an intermediate, that's like a product, a molecule that is created that is going to undergo some type of reaction to make another molecule. The enzymes are the proteins that catalyze this reaction, this transformation, okay? So for each one of these steps, there is a specific enzyme that is responsible for catalyzing this reaction. And remember that enzymes are specific for their substrates. The substrate is what you're starting with. All right, so let's look at glucose. First things first, glucose is what we call a hexose. It contains 6 carbons and it is a sugar. So let's count the carbons. There's 1, 2, 3, 4, 5, 6. It is a 6 carbon containing sugar. So we say it is a hexose. Hex for 6, ose for sugar. This is a hexose. Now, in the first step of glycolysis, glucose is going to be phosphorylated. Phosphorylation simply occurs when you add a phosphate group to something, to some compound. It can be added to a substrate. It can be added to another compound. But when you add a phosphate, you are phosphorylating something. And when you take a phosphate away, you are dephosphorylating. One of the enzymes that is instrumental in phosphorylation are kinase enzymes. Now, in step 1, the enzyme is a hexokinase. Whenever you hear kinase, you need to think about an enzyme that is going to be catalyzing the reaction that is moving a phosphate group from one thing to something else. In this case, I am going to be needing the phosphate, one of the phosphates found in ATP, to phosphorylate the number 6 carbon in glucose. So the phosphate needs to move from ATP to glucose. The enzyme that helps with this reaction is hexokinase, because glucose is a hexose and kinases move phosphates from one molecule to another molecule. This is the first step, and this is also the first ATP molecule that will be used in the energy investment phase. So your first molecule is used of energy. When this happens, I no longer have simply glucose. I have a glucose with a phosphate group attached to the number 6 carbon. So now the name of this molecule is not glucose, it is glucose 6-phosphate. So my substrate is glucose 6-phosphate. Now, glucose 6-phosphate is going to be changed into an isomer. We know glucose has 2 other familiar isomers, fructose and galactose. Glucose is an aldose sugar, aldehyde, and fructose is a ketose sugar, ketone. So glucose is going to be changed into an isomer of fructose in our next step. The enzyme, the class of enzymes that is instrumental in changing a compound into its own isomer is called an isomerase. Now, this isomerase is specific because it changes glucose 6-phosphate into its isomer. So The name of this isomerase is phospho for the phosphate, gluco because it came from glucose isomerase. The only difference between the second substrate and the third one is this is glucose and this is fructose. The reason this is done is because you see how this carbon is phosphorylated, the number 6 carbon is phosphorylated. I need to get my number 1 carbon in a position where it can also be phosphorylated. So now I have carbons 1, 2, 3, 4, 5, and 6. There is a phosphate on carbon 6, but I also need to add a phosphate to carbon 1. Think for a second, which type of enzyme is responsible for adding phosphates to something, taking it off of one compound and adding it to another one? Okay, if you said a kinase, you're correct. Now, I have another kinase, but this kinase works with fructose 6-phosphate. So therefore, its name is phosphofructokinase. Where did the phosphate come from? Another ATP molecule. Phosphate will be removed from ATP and added to the number 1 carbon on the fructose 6-phosphate. When this happens, I no longer have fructose 6-phosphate because this molecule not only has a phosphate on the number 6 carbon, it has a phosphate on the number 1 carbon. So now I have a fructose 1,6, meaning a phosphate on the number 1 and a phosphate on the number 6. This, because there's 2 phosphate groups, phosphate. My new substrate is fructose 1,6-bisphosphate. Now, I'm at near the end of my energy investment phase. There's only one more step. My last step involves breaking this 6-carbon compound, because there's still 6 carbons here, into 2 3-carbon compounds. So this is the step where this 1 compound has to be split into 2 compounds. This is done with the aid of an enzyme known as aldolase. The aldolase is going to assist with changing fructose 1,6-bisphosphate into 2 3-carbon molecules, G3P, or glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate, DHAP. G3P and DHAP are isomers of each other. They're both 3-carbon containing compounds. They both are phosphorylated on one of the carbons. So these 2 molecules are interchangeable. And the enzyme, again, that can turn a compound into its own isomer is an isomerase, a triose isomerase. Triose because these are 3-carbon compounds. Now, this is what I say about these 2. Glyceraldehyde is your go molecule. In order to go into the energy yielding phase, you need glyceraldehyde. DHAP is the wait molecule. So, if this process is ready to proceed, the molecule of DHAP will be changed to glyceraldehyde 3-phosphate, giving me 2 G3Ps. If it is not, G3P can be changed to DHAP and wait until the process is ready to proceed. At this point, I have used 2 ATP, and I've taken 1 6-carbon glucose and broke it down into 2 3-carbon molecules, G3P, DHAP. As I move into my energy payoff phase, this is the last step from the energy investment phase. Everything that happens now is going to be happening twice because remember, this molecule will become G3P and both of these G3P will go through the process. The first step is glyceraldehyde 3-phosphate needs to be oxidized. And when I oxidize G3P, NAD is going to be reduced to NADH. Nicotinamide is going to be reduced to NADH. Another thing that is going to happen at the same time, free-floating inorganic phosphate is going to be incorporated into this. Now, I'm not using a kinase this time because I'm not taking the phosphate off of another molecule. This is free-floating inorganic phosphate that's added in. Because I am reducing nicotinamide, I'm going to be taking some hydrogens from G3P. So this process is dehydrogenating. So the enzyme that aids with that is called a dehydrogenase. G3P is a triose and it contains phosphate. So the specific dehydrogenase is a triose phosphate dehydrogenase. When G3P is dehydrogenated, an inorganic phosphate is added in, I have what's known as 1,3-bis-phosphoglycerate. 3-carbon compound. On the number 1 carbon and the number 3 carbon, I now have a phosphate group. Remember, this is happening twice. It's going to happen to both G3Ps. So at this point, I'm netting 2 additional reduced NADH. 1,3-bisphosphoglycerate is going to give up a phosphate to an ADP molecule. Because I'm taking it from one compound and adding it to another one, my kinase is involved. This kinase is called a phosphoglycerokinase. And this is where I get my first molecule of ATP created. When 2 molecules of G3P go through, I'm going to get 2 ATP, 1 for each. Because I have removed the phosphate from the number 1 carbon, I no longer have 1,3-bisphosphoglycerate. I now have 3-phosphoglycerate. Remove the phosphate, change the name. 3-phosphoglycerate is going to be mutated. I went too fast. 3-phosphoglycerate is going to be mutated with a phosphoglycerate mutase. And simply what's happening is the phosphate is going to change from position 3 to position 2. So when this happens, I no longer call it 3-phosphoglycerate. It is now 2-phosphoglycerate. Phosphate moves from carbon 3 to carbon 2. Enolase is an enzyme that is going to remove water. When that water is removed, I now have what's known as phosphoenol pyruvate, or PEP for short, PEP for short. Phosphoenol pyruvate is the immediate precursor to pyruvate, the molecule I'm trying to make this entire time. Pyruvate is made by removing the phosphate group from the phosphoenol pyruvate. The phosphate is removed from this compound, added to ADP with help of a kinase, pyruvate kinase specifically. And I finally, after both G3P molecules have gone through this process, end up with my 2 pyruvate molecules. The 2 pyruvate molecules were formed at the end of the energy payoff phase. At the end of this phase, I've made 4 ATP, but I used 2 in the first phase. So we net a total of only 2 ATP. The pyruvate from the glycolysis is going to be oxidized to oxaloacetate. It's going to be oxidized to acetyl-coenzyme A. I apologize. Acetyl-coenzyme A is the key to Krebs. By binding to oxaloacetate, it creates citrate. I apologize, my system shut down. Now, as I was saying, when acetyl-coenzyme A bonds with oxaloacetate, it makes citrate, hence the name citric acid cycle. Citrate is going to undergo isomerization to become is isomer isocitrate. Now, isocitrate, and each one of these steps is being catalyzed by an enzyme. I'm only giving you the names of the substrates. Isocitrate is going to undergo transformation to become alpha ketoglutarate. To go from isocitrate to alpha ketoglutarate, carbon dioxide must be removed. So one of your original carbons is removed, and NAD again will be reduced to NADH. When you're left with alpha ketoglutarate, that same process is going to happen again. NAD is going to be reduced to NADH and carbon dioxide will be removed. And this is the last original carbon that came in from the glucose molecules. This carbon that is released as carbon dioxide, you make another molecule of nicotinamide that is in its reduced form, and that leaves you with succinyl coenzyme A. Succinyl coenzyme A, going the wrong way, is going to be transformed to succinate. Now, with the use of GTP, guanidine triphosphate, and inorganic phosphate, the removal of coenzyme A, ATP is going to be created through a phosphate level, substrate level phosphorylation. So this is where you get your one molecule of ATP. Succinate will be transformed to fumarate. That process is going to remove 2 electrons. So in this process, this is where your flavin adenine dinucleotide is going to be reduced to FADH2. This is the only place where you get your FADH2. Remember F and F. Fad before fume. Fad before fumes. All right. After fumarate, water is going to come in. Fumarate becomes malate. And the last reduction of nicotinamide happens as malate is formed back into oxaloacetate, thus completing the cycle. So you're going to get your nicotinamides being reduced at steps 3, 4, and 8. Fat is reduced at step 6, and ATP is made at step 5. Now, this is showing the pathway of electron transport. What you have here are your multi-protein complexes. You have protein complex 1, 2, 3, and 4. Remember, NAD drops off at 1, FAD drops off at 2. FAD is not an integral protein. It is attached to the plasma membrane, but it does not embed through it. So as these electrons are being brought to the multi-protein complex, they're transferred from NAD and FAD to the electron transport chain. And these chain components contain, some of these contain iron in these atoms or whatever. This is why iron is important for us. Now, if you're anemic, you'll see the results of that. Because if you're anemic, a lot of the symptoms tends to be that you're tired. And I'm probably oversimplifying this, but if you think about ATP as cellular energy, if I can't make ATP, there's no energy. Lack of energy equals tired. That's how that plays in. The carriers are going to alternate between reduced and oxidized states as they accept and donate electrons. And the final electron acceptor in this process is going to be the oxygen molecule. And when oxygen accepts the electron, it is going to bond with protons to make water as a byproduct. Now, keep in mind that my PowerPoint is still not working. But the key thing to remember is that during electron transport, no ATP is going to be directly generated. Electron transport does not directly make ATP molecules. So, why is it important that the electrons are actually passed down in the electron transport chain? Again, we are trying to control the change in free energy. It breaks down that large free energy drop from food to oxygen. And by doing it in smaller steps, that release happens in a manageable amount. Now, chemiosmosis is the energy coupling mechanism. And so chemiosmosis is going to have the electron transfer and electron transport driving it, because it is going to cause protons to be pumped out by these proteins into the mitochondrial matrix, from the mitochondrial matrix into the inner membrane space. The protons actually move back into the matrix through this ATP synthase, which is a membrane protein complex. And by doing that, it turns, there's a rotor protein in there, and those protons are instrumental in turning that rotor protein, that catalytic knob. And when that happens, the exergonic flow of electrons, so the energy releasing flow of those protons, I'm sorry, I said electrons, the energy releasing flow of those protons is going to drive the phosphorylation of ADP to ATP. So this is how electron transport basically creates a proton motive force, and this force is going to drive the phosphorylation of ATP by having an exergonic flow. This is an example of chemiosmosis, because it's the use of the proton gradient to drive cellular work. Here is an overview of glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. So essentially what you have is, in glycolysis, glucose is going to be split into two pyruvate molecules in the cytosol. That process generates a net of 2 ATP and 2 NADH molecules. Those NADH molecules will head towards the oxidative phosphorylation stage. The pyruvate molecules will enter into the mitochondria, undergo oxidation to become 2 acetyl coenzyme A molecules. That process releases carbon dioxide as a byproduct and also generates 2 NADH. Those NADH will also move on to the oxidative phosphorylation process. Acetyl coenzyme A is the key that turns Krebs. After those 8 steps of the Krebs cycle, where pyruvate has been turned to acetyl coenzyme A binds to oxaloacetate to make citrate. Citrate is changed to isocitrate, then alpha ketoglutarate, then succinyl coenzyme A, then succinate, then fumarate, then malate, and back to oxaloacetate. For each one of these that enters, you're going to have a total of 3 NADH, 1 FADH, and 1 ATP. Since this process happens twice for every glucose molecule, we're going to net 6 NADH molecules, 2 FADH2 molecules, and 2 ATP. Those NADH molecules and FADH molecules are taken to oxidative phosphorylation, which happens in the cristae, the folds of the inner membrane of the mitochondria, where those hydrogens are stripped of their electrons to use in the electron transport and passed to oxygen, the final electron acceptor. The protons are used to create a proton motive force and a proton gradient across the membrane, which drives chemiosmosis by re-entering the membrane through ATP synthase. The most ATP you can get from oxidative phosphorylation is about 26 or 28 ATP. So per glucose molecule, you're going to maximize at 30 to 32 ATP. Proteins, carbohydrates, and fats, remember, can all undergo catabolism. They can all be broken down. But they enter this process at different stages. Your carbohydrates or your sugars are going to enter at the top, specifically glucose. Your proteins tend to enter around the end of glycolysis or during pyruvate oxidation or directly into the citric acid cycle. And your fats tend to enter around the glyceraldehyde 3-phosphate stage with the glycerol parts, because remember, your fatty acids are 3 carbon sugars and hydrocarbon chains. So the 3 carbon sugar, the backbone, will enter after the energy investing phase, right before the energy yielding phase, because it is already a 3 carbon sugar. And your fatty acids will enter around the process of pyruvate oxidation. But again, your body will burn carbs first, lipids second, proteins last. I hope this has been helpful.